Good morning, everyone. Welcome to the September Zoom Citrus Workshop. Thank you for joining us. First, I would like to thank Alison Walton from the Immokalee IFAS Center for her help and cooperation. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. Today's program will focus on evaluating Brassinosteroids to improve citrus fruit quality and tree protection under HLB. Our guest speaker is Dr. Fernando Alferez. He is an associate professor in citrus horticulture at the UF-IFAS Southwest Florida Research and Education Center in Immokalee. Before Dr. Alferez gives his presentation, I will go through some comments related to Brassinosteroids. Brassinosteroids are growth hormones that are found in all plant species. Brassinosteroids are a class of novel plant hormones gaining importance in agriculture. Brassinosteroids are products that control plant growth and development. Their use is an important strategy to increase crop yield and improve crop quality. One of the main goals of global agriculture is improving the yield and quality of crops. This goal can be achieved by improving the ability of crops to resist biotic stresses, such as pathogens, and by improving the ability of crops to resist abiotic stresses, such as drought and cold. Brassinosteroids are plant growth regulators that improve plant immunity, promote plant health, and reduce the damage caused by adverse weather conditions to crop. For citrus, use of natural brassinosteroids can accelerate nutrient absorption and utilization, promote cell division and fruit development and growth. At the cellular level, brassinosteroids are involved in increasing chlorophyll content, photosynthesis, carbon fixation, and sugar content. At the plant level, brassinosteroids have been shown to improve growth and development, enhance fruit yield and quality, stimulate the natural defense mechanisms of plant, and reduce disease pressure. They are also involved in salt, cold, heat, and drought tolerance. You are all informed and know about IPCs. Individual protective covers have been found by Dr. Alferez and his team to be an effective tool in protecting newly planted citrus trees from HLB. However, this protective system has to be removed after 2 to 3 years, leaving trees exposed to the psyllid vector of the citrus greening disease. Adding brassinosteroids treatment when IPCs are removed may prolong tree health and improve fruit yield and quality. Again, our guest speaker is Dr. Fernando Alferez. He will give an overview of his work with brassinosteroids in the past 5 years. He will show how brassinosteroids can protect young citrus trees from HLB, and he will show how Brassinosteroids can improve citrus fruit quality. He will also present work currently being done on using Brassinosteroids to alleviate some side effects that OTC injections may have. It's all yours, Dr. Alferez.
Thank you, Mongi. Thank you very much for this nice introduction. And I want to thank also Alison Walton for her great assistance with Zoom this morning. Your help is greatly appreciated, Alison.
Thank you, Dr. Alferez.
Thank you. So I will start just with an overview of what we are going to talk about today. And I will start with some introduction on the current situation that we have now in Florida. Then some notes about Brassinosteroids that were kind of pointed out already by Mongi, but I will give some more details. And then I will focus on the research that we are doing on young trees, and then on mature fruit-bearing trees. And then I will finish with some new research directions that my program is taking in both young and mature trees. And this is ongoing work, but I will show you some interesting data already. Okay, so as you know, in my program we are working basically with both mature and young trees, and this is for a reason. Now, this is the situation that we have now in Florida. So we have a lot of mature trees that are already affected by HLB, and they are declining in production. And at the same time, we are planting new trees in the ground that are healthy from the nurseries, and they are not producing yet. So the biology of these two types of trees is totally different. So they have different requirements. But the desired goals for both are in the case of mature trees to maintain them productive and, if possible, improve their health, and of course improve fruit yield and quality when possible. And for the young trees, of course, keep them free from disease until they enter into production age or longer if possible. Okay, so in general, this is the dire situation that we have now in Florida when we plant a new growth. According to our data, if trees are not protected by 6 months, we have about 80% of the trees HLB positive, and by the first year, all trees are infected. Okay, now let me talk to you a little bit about Brassinosteroids. As Mongi said, there are a relatively new class of plant growth regulators. They were discovered about 40 years ago. And the nice thing about them is that they have a myriad of interesting effects in plants. So now they have been reported to increase stress tolerance, both biotic and abiotic. They also induce photosynthesis. They can modulate plant architecture and branching, which is really interesting, and also improve root development. And very interestingly for us, when we started this work, there have been associated with resistance to some diseases in other plants. But there was a report from Cuban researchers a few years ago showing that Brassinosteroids could actually reduce psyllas titer in citrus trees. And also there are some reports on fruit maturation advancement in strawberry, a fruit that, like citrus, is an non-climateric fruit. So, and coincident with this, there was a report a few years ago, 3-4 years ago, showing that in blood oranges, Brassinosteroids, a treatment with Brassinosteroids can maintain sugar levels in use, okay, as opposed to natural reduction during post-harvest storage. Now, how all these pieces of information that I just presented could help us in the situation that we have. As Mongi introduced, we now know that individual protective covers can maintain trees free from disease, free from HLB, with no bacteria detected in the protected trees, okay? And with improved growth. As you can see in the picture here, a tree that was covered with IPCs as compared to a tree that was not covered with IPC for about 3 years, okay? Now, the problem is that this effect disappears as soon as we remove the covers, and the trees start to get infected as they are now exposed to psyllids and to the environment. So we started a trial treating newly planted trees with Brassinosteroids, taking into account all the information that I shared in the previous slide. And we were treating the trees once per month with a concentration, and this was really interesting. We did a lot of trials with different concentrations and different timings, and we were trying at this time monthly with 6.2 ounces of product in 100 gallons of water, and that is what we are basically doing now. And we found a clear delay in the infection rate. So by 6 months of treatment with Brassinosteroids, only 25% of the trees were testing positive for HLB, as compared to 80% of the trees already infected by these 6 months in the non-treated trees, in the control trees. And also we found and we saw some improvement in growth, as you can see in this picture. Here you can see 2 controls that are clearly different in size and in volume as compared to the trees that were treated with Brassinosteroids that were more dense in canopy and more uniform, and that was really consistent across the whole trial. Okay. So this basically what takes me is to the work that we are doing on young trees, and I will start now talking about the work that we are doing in young trees. Okay, and we applied and we got a grant from USDA NIFA a couple of years ago for studying the combination of IPCs and Brassinosteroids as an integrated strategy for maintaining young trees free from disease and healthy, well into the productive age, which is our main goal. So basically what we did is we planted the trees in January 2020, and we maintained the trees with IPCs for about 3 years, okay? And then we either left them exposed to the infection, or so we removed the covers, and we left them exposed to the environment, or we removed the covers, but we treated monthly with Brassinosteroids at the rate that I just mentioned before, okay? So we are looking into all these parameters here, like bacteria titer, the psyllid population, any other insects or diseases that are present in these trees, how the canopy is developing, how is the tree growth, how are the flushing and blooming patterns, if we have fruit, how is the fruit set, and fruit yield and quality, of course, and also some other physiological and biochemical indicators that I will mention a little bit later today. Okay. So we have now several trials in both our experimental farm here at Southwest Florida Research and Education Center, and also with a couple of commercial collaborators. And these trials involve different types of mandarins, sweet oranges, and pomelos. And I'm going to show some of the data that we are getting so far. And again, this is an ongoing project. We are in the equator of the project as we speak. Okay, so I'm going to show some data on tango mandarins, and this is how tango trees look after we remove the covers. Okay, and we treated with Brassinosteroids for 6 months. On the left, you have a tree that was covered for 3 years and not treated. So 6 months after we remove the cover. And the tree looks nice. It has a nice canopy, and it has a nice number of leaves, and it looks basically healthy. But compare that with the tree that we were treating for 6 months, once per month with Brassinosteroids. So you can see more flashes and a denser canopy. Besides, okay, the flashes were more abundant, but they were also earlier and more synchronized. And as a result, we had a denser canopy with greener leaves, more chlorophyll content. We found more fruit set in these trees. And most of the trees after 6 months were still HLB negative. Now the rootstock was thicker, and the volume of the canopy was larger. Now we have seen, and we're still working on this, but we have seen some new phloem vessels in these shoots as compared to these trees. So there is some phloem actively being generated in these trees under Brassinosteroid treatment. We are finding less callose deposition. Remember, these trees are mostly HLB free. And we have seen also an increase in enzymes related to oxidative stress alleviation. So what I'm saying here is that the effects that we are seeing on with the Brassinosteroid treatment are really widespread. There is, as I showed in one of my first slides, that basically combine what we knew about different model plants. We are seeing a lot of these effects in our treatments with Brassinosteroids. Okay, and one of the things that we found that was very interesting is that we found a synchronization in flushing, as you can see in this graph. Okay, flushing started earlier with Brassinosteroids because when most of the flushes were already fully expanded, in the non-treated trees, we still had some feather, and most of them were still at expanding stage. Okay, and this may have some relevance as flushing synchronization may be of great interest for psyllid management, for instance. So if we can advance and synchronize flushing, that can be a very interesting tool for us. Okay, and we also had some very interesting data on tree phenology, and here I show the data at the time of IPC removal, okay, and then at 6 and 12 months after removal and monthly treatment. Now I want you to focus. Well, we have all the treatments here, so the blue boxes are no IPC from the beginning and no Brassinosteroid treatment, so control. Then in the purple, we have no IPC from the beginning, but then after the 3 years in the ground, we started the Brassinosteroid treatment. Then the pink boxes are the trees that were covered for 3 years, and then we removed, but we didn't treat with Brassinosteroids, so we left exposed to disease. And then the green box is the trees that were covered for 3 years with IPCs, and then we removed the IPCs and we started the treatment with Brassinosteroid once per month. And And you can see how by 1 year after we removed the covers and we started the brassinosteroid treatment, we see a clear significant effect on rootstock diameter increase. We don't see the same thing for scion diameter, but still there is a trend, there is an increased trend. Canopy volume is largely improved by 1 year in the brassinosteroid treated trees that were covered with IPCs, as you can see here. And probably as a consequence of this, we got a much better yield in these trees, because remember, yield is a direct consequence of canopy volume and number of leaves. I remember a classical professor in Valencia, in Spain, in the Polytechnic University, José Luis Guardiola. He always said that you need 33 leaves to make a fruit. So in this case, we have an abundant number of leaves, and that results in more yield. Okay, so our conclusion here is that IPCs confer a significant advantage to trees even 1 year after removal, as we can see here. But brassinosteroids are able to enhance this advantageous effect of IPCs. Well, when we looked into the HLB infection rate, what we found was really interesting. You can see how at the time of IPC removal, the trees were free from disease, okay? Were free from HLB, the boxes pink and green, as they were above the cutoff of the threshold of 36 in city value. Well, I have to say that we had a tropical storm and some covers blown away, so that is why probably the green box has some values below 36. But look what happened after 6 months of treatment with brassinosteroids. The green box is well above the 36 threshold cut. Of course, the trees that were covered but not treated afterwards, the pink ones, okay, the pink box, started to get infected. And the trees that were not covered from the beginning, that were infected from the beginning, did not recover, even though we were treating them with brassinosteroids, the purple box, okay? And this is important because it suggests that a reinfection may be an important factor here, okay? So these trees are getting exposed to psyllids, so they are getting reinfected. So the treatment that we are doing with brassinosteroids is not helping these trees if they are infected and exposed. And I will talk a little bit more about that later. Now, we are interested now in understanding the real effects of brassinosteroids just because what I just mentioned, and what I mean is that we are interested in knowing which part is related, which part of the effect that we are seeing on brassinosteroids is related to induction of immunity, and what part is related to physiological effects per se. So, and for this, we are collaborating with colleagues in Valencia, Spain, because as you may know, so far, and crossing my fingers, Spain is, or Valencia is an HLB-free area. And so what we are doing is treating trees from the same age in both locations. So we are treating trees both in Valencia and here in Florida. They were planted at the same time. And we are treating with the same rate of brassinosteroids in both locations. And we are collecting the same physiological and phenological data. So we are hoping that this will help us to understand better the trade-offs that may be involved in the brassinosteroid response, as there is always a balance between immunity and growth in plants. Now, The trial in Spain is funded by the Spanish government, and this is a view of our trial in Spain. This is in southern Valencia, in Gandia. And here you can see a control tree. This is 1 year after planting, and here you can see a brassinosteroid treated tree at the same rate that we are doing here in Florida. And here in Florida, what we are doing is we are doing an experiment in the field. That's this one, but we are doing also an experiment in pots. Okay, and this is our pot trial in Florida. So we have about, I believe there are 90 trees, and this is also after 1 year. Okay, and you can see a clear difference also in tree growth here. And these trees, remember, they are exposed to psyllids and they are exposed to the disease here. And we last week or a couple of weeks ago already, we excavated some trees, and we took some samples and data, and I will show you some initial results. We are still processing all these samples and all this data. It's a lot of work. But here you can see, well, first of all, you can see the root mass and the trunk diameter in a control tree and in a tree that was treated monthly. You can see that the rootstock diameter is clearly larger than in the control, and we are still looking into the root mass volume, but we can see already some things, like for instance, much more new growth in the root masses that were from the trees that were treated with Brassinosteroids than in the controls. Actually, in the controls, we can see some decline in the root system. And also something really interesting for us was that lateral branching was really induced with Brassinosteroids. And you can see that it was significant in the treatments that we were doing every other week or once per month. Not so in the trees that we were treating every other month. But also very interesting, the number of fruits that we found per tree. In this case, because they are small trees, we did the count per each plot, that are plots of 4 trees, and in control we didn't find any fruit. There was no fruit set, and we didn't find any fruit. And you can see how the trees that were treated with Brassinosteroids were setting and developing fruit in their canopies. Now, this is a project that is continuing, and we are really encouraged with these results. Now, changing gears now, I'm going to talk about the work that we are doing on mature fruit-bearing trees. And we have been working now for 2 years in a project funded by CRDF in Hamlin and Valencia to try to understand how Brassinosteroids may advance fruit maturation, and we are getting some very interesting results. So the first year, this is the first year that we did the experiment, the trial in Hamlin. And the first year we were able to show that it only takes 1 application of Brassinosteroids at the right time to increase Brix by about 1 degree. And in this case, it was somewhere or sometime during November, okay, between early and mid-November. Then we have a response, okay? And we got also a modest increase in yield, and this was in 2022. And the same was true for Valencia, okay, in that season. 1 single application, and that was between mid-February and early March, increase bricks significantly and put bricks well above 9 as compared to the controls, okay, that were in around 8. Okay, the same for the yield. We got a mild effect on yield. Now, we analyze in these samples here, the samples that we collected in March. We analyzed sugar content and acid, organic acid content at that time, and we found a significant increase, for instance, in fructose, okay, when we treated with brassinosteroids. And it was about 45% more than in controls. And then an increase in acids like citric acid. Some other sugars will also increase, even though they were not significant, but look at glucose, for instance. Okay, there was a trend there. Okay, and this was really encouraging. So last year what we did was to repeat the treatments and the trials. And here comes the interesting thing. So this is what we found in Hamlin. In Hamlin, we could not get any difference in bricks, acidity, or ratio. Okay, everything looked basically the same with no significant differences. And the same results we got in Valencia. No significant differences and no response to brassinosteroid treatment when we were looking into the data for all the trials. Okay, so we were puzzled for a while about these results. Fortunately, we have huge trials with a big number of replicates, and the student who is doing this work, my PhD student, Divya Aryal, she keeps really good track of every single tree and records every single tree and analyzes fruit from each replicate tree individually, even though we later may pull the data. So what we did was just to look into the tree health parameters of each individual tree, as in the scale that we normally use. Remember that this scale goes from 0, which are really healthy trees, to 5, which are heavily declining trees. Okay, and we found that most of our trees in the first season were at a score of 2 or 3, as in this picture. And in the second season, there was a clear decline in that growth, and most of the trees were mostly at 4, as I show in this picture. So we came back to our data, and I will show you just an example in Valencia, because we have multiple examples, but I will just show one in Valencia. And so we selected a data point in which it looked like for last season, there was some trend in bricks increase after our treatment. This is the blue box, okay? And then we organized the data, the bricks data actually as related to tree health. So we pulled the trees and we separated the trees in trees that were scoring a 4 and trees that were scoring a 2 or a 3. And in that case, we saw an effect. So trees that were at a score of 2 or 3 had a significant increase in bricks with the brassinosteroid treatment, as compared to the trees that were at 4, so the declining trees. So this clearly indicates that for these treatments to have a significant effect, so we can see something in bricks increase, we need trees in the best possible shape. And this takes me now to share with you some of the things that we are planning in my program and starting to do now. And the first thing is, can we improve fruit quality in trees that are treated with oxytetracycline? As you all know, now oxytetracycline is a treatment that most of the growers are doing because it's showing really good results with tree health and with fruit yield and quality. So in this case, as I show in this picture, which is from a paper by the group of Dr. Ute Albrecht, what I'm showing here is a control tree and a tree that was treated with oxytetracycline with a clear recovery of the canopy and the general tree health. So in this case, can brassinosteroid applied here to improve even further quality of the fruits that are growing in these trees? Because these trees are in the scale, they are probably about 0 or 1 in the tree health scale. So we are working on this right now, and we are starting some probably between this and next month some treatments in trees that have been already injected with oxytetracycline to see if a treatment with brassinosteroids can increase or improve fruit quality. Okay, this is one area that we are actively pursuing this coming season. Now, the second area is to use brassinosteroids as an aid treatment. Okay, there are some reports that, or I heard some reports showing that OTC treatment, even though is resulting in tree recovery and increased fruit production, may be related to an unexpected increase in canker incidence, okay, to some extent. The problem is that treating these trees with copper to avoid canker incidence, at least with the current formulations that we have, may result in an increase in rust mite incidence, okay, among other problems. Now, we are now assaying with my collaborator, Dr. Suarez Santra in University of Central Florida. We are assaying a combination of novel copper formulations and brassinosteroids, so we can tackle this problem from different points. Okay, on one hand, brassinosteroids may help to increase yield and fruit production, and also fruit quality. And they may also, and we have some data showing that may reduce rust mite incidence, okay, when rust mite is present. And that's on one hand, but on the other hand, novel copper formulations, we are hoping, or we believe that they will be abating canker incidence, and we are hopeful that we will get some funding for this project soon. And the last idea that I want to share with you is to see if we can recover infected young trees from HLB by using brassinosteroids as a therapy. Okay, in other words, imagine that you have planted trees without protection, and the trees are now growing and are HLB positive after 1 year in the field. So the question is it too late for these trees? Can we do something here? And to answer this question, I started a trial last year with 90 Valencia trees grafted on US 942. and they were planted in February 2023, and they were non-protected. Okay, so they were left exposed to psyllids and other pests and diseases. And in February this year, so after 1 year in the field, all the trees we tested and they were HLB positive. Then we did three things. Okay, so what we did was first keep 1/3 unprotected, then cover another third of the trees with IPCs, and then the last third we covered with IPCs, and then we started treat them with Brassinosteroids once per month. Okay, and we are still working on this, and it's been 6 months now. But look at this picture. So this is after 6 months, and it looks clear that, well, this is a tree that has been now 18 months exposed. Okay, this is the untreated control. So this is what we are getting when we put the covers, the IPCs on the trees, and this is 6 months later. So we're avoiding any reinfection, and what we see is more leaves in these trees, so the canopy is denser. But look what happened when we when we cover the trees with the IPCs and we start treating with Brassinosteroids. So this has been 6 applications of Brassinosteroids. So we are getting a more denser canopy, greener leaves, and the canopy is really expanding really well. So it's clear that Brassinosteroids in this case are improving growth, and we are continuing this trial for at least 1 more year. So I will be informing on the outcomes of this trial and others that we may be planting in the next future in terms of bacteria titer, tree growth, and eventually fruit production in all 3 cases. So we will have a clear idea of what happened and if we can recover these trees that were not planted protected at the beginning, with protection at the beginning. So this is basically all what I had to share with you all today. So, and this is a summary. So we can say that Brassinosteroids, we have shown that they improve growth, they improve branching, and immunity if applied to young, less than 3-year-old trees. Now, to mature trees, if we apply Brassinosteroids at the right time, we may be able to improve internal fruit quality and to increase Brix and especially the ratio. Okay, but this effect of Brassinosteroids in mature trees is mostly significant if the trees are at 3 or less in the scale of HLB damage. So there is a strong potential here for their use in combination with OTC-treated trees, as these trees that we have been treating with OTC show signs of recovery and improved health. And it seems that Brassinosteroids may recover trees that are already infected or affected by HLB if no reinfection occurs in those trees. So if we protect the trees. And that's all I have. Just I want to thank my excellent team for all the great work they are doing. Osvaldo, Maria, Divya, Susan, Murillo, and Pablo. And to my international collaborators, Drs. Perez, Edo, and Urbaneja from Valencia, Spain, and also to our shared PhD student, Raúl Hortels, who regularly visits our lab, and of course to our funding agencies for their trust in what we are doing, USDA, IFAS, CRDF, and also the Spanish Ministry of Science. And that's all I have. Thank you very much. And I will take any questions that you may have.
Any question or comment?
Dr. Alferez, there is a question in the chat. It says, which is the best time to apply BR on Valencia? And then which other hormone do you recommend to apply together with BR, since the label of Brassinoid
Yeah.
is 0.1% requires it?
Yes. The right time for Valencia is it's going to depend on the area that you are. But I would say is after the main bloom. Okay, is somewhere between late February and March. Okay, and mid-March, in that window. I know of cases that have been applied in December or in January, and still they got really nice results in bricks increase. But my experience is that we get the best results when we apply in mid-February. Now for application with other hormones, actually the label of one of the products that we are using, that is produced by Repar, they are suggesting to apply with gibberellic acid. I have not done that, and I am planning to do that this coming season. And I will have some more information about that in the next future. To me, it's an interesting approach, but I have no data on that, on what can be applied at the same time.
Any other question? If no question, then thank you very much, Dr. Alferez. And again, if you need CEUs, email me your name, email address, and license number. Thank you all for attending, and see you next time.